Hello, everyone. I welcome you once again to another wonderful time with Dr. Ifeanyi Mary Ann Okafor. Thank you for being part of Hematology and Blood Transfusion Science Lecture Series. Thank you for subscribing to our YouTube channel. For some time now, we'll be handling various topics. We've treated iron metabolism, iron absorption, regulation of iron deficiency anemia. We've talked of vitamin B12 metabolism, folate metabolism, megaloblastic anemia. We've also handled topics on blood group system. Please, if you are interested in any of these lectures, visit our YouTube channel and listen to them. I bet you are going to learn a lot from them. And if you have any question in any of the topics that we have handled so far, don't hesitate to send in your questions. You can put it on the comment section of our YouTube channel, or you can also send it to our email address, scholarlywoman20@gmail.com. Thank you very much. And if you have not subscribed, this is a great opportunity for you to do so. You can hit the button now. Please keep us sending in your feedback. It's actually helping us a lot to get better. And also, don't forget to like our videos and comment. Thank you very much. Today, we are going to be handling another exciting topic, hemoglobin synthesis and catabolism. Hemoglobin synthesis and catabolism. Let us go straight to today's lecture. See you at the end of the lecture. Today's topic, hemoglobin synthesis and catabolism. I am Dr. Ifeanyi Mary Ann Okafor from the Department of Hematology and Blood Transfusion Science, Faculty of Medical Laboratory Science, College of Medical Sciences, the University of Calabar, Nigeria. It is expected that at the end of this lecture, you should have a good knowledge of how hemoglobin is synthesized, the processes that are involved in hemoglobin synthesis, and you should be able to explain hemoglobin catabolism. The main function of the red blood cells is to carry oxygen to the tissues and to return carbon dioxide from tissues to the lungs. And that is the primary function. In addition to any other function that the red cell can perform, the primary function of red blood cell is to carry oxygen to the tissues from the lungs and also to return carbon dioxide from the tissues to the lungs. In order for the red cell to achieve this gaseous exchange, it contains a specialized protein called hemoglobin. It contains a specialized protein called hemoglobin. Each red cell contains approximately 640 million hemoglobin molecules. Each red cell contains approximately 640 million hemoglobin molecules. Now, hemoglobin is a red globular protein found inside the red blood cells, which have a molecular weight of about 68,000 daltons and comprises almost 1/3 of weight of a red cell. It is a red globular protein that is found inside the red blood cell. And it comprises almost 1/3 of the weight of the red cell. Now, hemoglobin has two basic components, as shown on the slide here. Hemoglobin is made up of two basic parts, the HAEM part and then the globin part. The HAEM part and the globin part. Hemoglobin is made up of two basic components, the HAEM part and the globin part. About 65% of the hemoglobin is synthesized in the erythroblast. About 65% of the hemoglobin is synthesized in the erythroblast, and 35% at the reticulocyte stage. If you remember your lecture on erythropoiesis, and then you should be able to know stages that are involved in red cell production. You should be able to know when we say erythroblast, you should be able to know the stage we are referring to. When we say reticulocyte stage, you should be able to know the stage we are referring to. So 65% of hemoglobin synthesis occurs in the erythroblast, while 35% occurs at the reticulocyte stage of erythropoiesis. HAEM synthesis occurs largely in the mitochondria. HAEM synthesis occurs largely in the mitochondria, while globin synthesis occurs in the polyribosomes that is present in the cytoplasm. Globin synthesis occurs in the polyribosomes that is in the cytoplasm. HAEM synthesis occurs largely in the mitochondria. Note, although HAEM and globin synthesis occur separately within developing red cells, as I've just said earlier, HAEM synthesis occurs largely in the mitochondria, while globin synthesis occurs in the cytoplasm of the cell, in the polyribosomes. So although HAEM and globin synthesis occur separately within developing red cell precursors, their rate of synthesis are carefully coordinated to ensure optimal efficiency of hemoglobin assembly. Although HAEM is produced largely in the mitochondria and partly in the cytoplasm, and globin synthesis occurs in the polyribosome in the cytoplasm separately within the developing red cell precursors. The rate of their synthesis are so synchronized and coordinated, that there is optimal hemoglobin assembly. That is, once the HAEM is being produced, the globin at the same time also being produced at the same rate, and at the end of the day, both is assembled and hemoglobin at the end of the day is formed. We are going to take a deeper look on this as we go on in the lecture. Globin synthesis. The various globins that combine with HAEM to form hemoglobin are all single-chain polypeptides. The various globins that combine with HAEM to form hemoglobin are all single-chain polypeptides. The synthesis of these globins is under genetic control. The synthesis of these globins is under genetic control. Humans normally carry 8 functional globin chains. Humans carry 8 functional globin chains arranged in 2 duplicated clusters. Human normally, remember we are discussing globin synthesis, and we said that various globins that combine with HAEM to form hemoglobin are all single-chain polypeptides. And the synthesis of these globin are genetically what? Controlled. And humans carry about 8 functional globin chains arranged in 2, and 2 duplicated gene clusters, alpha-beta cluster, that is responsible for coding for the genes like beta, gamma, delta, epsilon globin genes. And these genes are located on the short arm of the chromosome 11. And the alpha-like cluster that is responsible for these genes, alpha and zeta globin genes, that's found on the short arm of chromosome 16. Beta, gamma, delta, and epsilon globin chains. The genes responsible for these globin chains are found on the short arm of chromosome 11. And then the gene responsible for alpha and zeta globin chains, that is alpha and beta globin genes, are found, the gene is actually found on the short arm of chromosome 16. The alpha-like cluster is found on the short arm of chromosome 16. Globin component of different normal hemoglobin. Now we have different normal hemoglobin depending on the stage of the human being. Remember the basic function of hemoglobin is to carry oxygen from the lungs to the tissue and also take the carbon dioxide from the tissue to the lungs. And depending on the stage of an individual, right from conception, we have different forms of hemoglobin that are engaged in carrying out this function effectively. So at embryonic stage, we have what we call embryonic hemoglobin. At the embryonic stage of the individual, we have what we call embryonic hemoglobin that are involved in carrying this oxygen from the lungs to the tissue. And remember we said hemoglobin consists of heme and the globin part. So whether it is embryonic hemoglobin, fetal hemoglobin, or adult hemoglobin, all of them have the heme and the globin part. So normal hemoglobin we have in individual, depending on the stage of the individual, we have embryonic hemoglobin that functions at embryonic stage of the individual. And then we have fetal hemoglobin at the fetal stage, and then the adult hemoglobin. All these are normal hemoglobin that function at various stages of an individual. As individuals grow from embryonic stage, we have 3 types of embryonic hemoglobin. Hemoglobin GOA1, and what we have by the side is actually the globin chain. Most times, the heme aspect of hemoglobin is constant, irrespective of the type of hemoglobin. What actually differentiates one hemoglobin from the other is the globin aspect of the hemoglobin. So in all these different types of hemoglobin, the heme part of the hemoglobin is constant, but the variation occurs in the globin chain. So at embryonic stage, we have 3 types of embryonic hemoglobin. Hemoglobin GOA1 that has the globin chain as zeta, 2 chains of zeta globin chains, and 2 chains of epsilon globin chain. In addition to heme group, we have hemoglobin Portland that has 2 chains of zeta globin chain and 2 gamma globin chain. And then we have hemoglobin GOA2, and we have that has 2 alpha globin chain and 2 epsilon globin chain. So these 3 types of hemoglobin is called embryonic hemoglobin. Hemoglobin GOA1, hemoglobin GOA2, and hemoglobin Portland. These are normal hemoglobin that are found at embryonic stage of an individual that carries oxygen from the lung to the tissue for the developing embryo. As the embryo is growing, is developing, that is a switch from embryonic hemoglobin to fetal hemoglobin. That is a switch from embryonic hemoglobin to fetal hemoglobin. And fetal hemoglobin, what we call hemoglobin F, has 2 alpha globin chain and 2 gamma globin chain. Fetal hemoglobin has 2 alpha globin chain and 2 gamma globin chain that make up the globin aspect of the hemoglobin, fetal hemoglobin. So it has the constant heme group and then the globin chain made up of 2 alpha chains and 2 gamma chains. And then we have adult hemoglobin. In adult stage, we have two types of adult hemoglobin, hemoglobin A and hemoglobin A2. Hemoglobin A and hemoglobin A2. The hemoglobin A, which is the predominant form of the hemoglobin that you see in adults, is made up of, the globin chain is made up of 2 alpha globin chains and 2 beta globin chain. 2 alpha globin chains and 2 beta globin chain. While hemoglobin A2 is made up of 2 alpha globin chains and 2 delta globin chains. 2 alpha globin chains and 2 delta globin chains. Globin synthesis is first detected in the primitive erythroid precursor of the yolk sac at about 3 weeks gestation. Globin synthesis is first detected in the primitive erythroid precursors of the yolk sac at about 3 weeks gestation. Globin chains are assembled by the cytoplasmic ribosomes. Remember, we said globin chain is synthesized in the ribosomes, polyribosome that is found in the cytoplasm of the cell. Globin chains are assembled by the cytoplasmic ribosomes under the control of 2 clusters of closely linked genes of chromosome 11 and 15. Like we said earlier, these genes are beta-like cluster genes that are responsible for the 4 globin chains, the beta, the gamma, the delta, the epsilon, and then the alpha-like cluster genes that found on chromosome 16 that is responsible for the alpha and the zeta. Globin messenger RNA. Globin messenger RNA sequences are first expressed in the pronomoblast and early basophilic erythroblast. That is the messenger RNA that is involved in synthesizing globin is first expressed in the pronomoblast and early basophilic erythroblast. And globin chain synthesis depends on accumulation of globin messenger RNA, increasing at polychromatophilic and autochromatophilic stage of erythropoiesis. This messenger RNA accumulates and increases as the red cell precursors are maturing, and at polychromatophilic stage and autochromatophilic stage of erythropoiesis. During the later stage of erythroid cell maturation, the amount of RNA per cell and the rate of total protein synthesis declines, but the relative stability of globin messenger RNA ensures that globin becomes the predominant polypeptide made in the late erythroblast and at the reticulocyte stage. Remember that during erythropoiesis, you know that one of the striking things in erythropoiesis is that as the red cell matures, the nucleus keeps shrinking until it exudes out of the cell. So you know that mature red blood cells do not have a nucleus. So another striking thing that occurs in red cell maturation, as red blood cell matures, the rate of hemoglobin synthesis also increases. And that is why you see here that during this globin synthesis, the messenger RNA accumulation increases even at polychromatophilic stage of erythropoiesis and autochromatic stage of erythropoiesis. And then at the late stage of erythroblast and at the reticulocyte stage, which is actually the last stage of red cell erythropoiesis prior to the fully matured red blood cell, that is the reticulocyte stage. At this particular stage, the predominant polypeptide is actually the globin chain that is already being produced, waiting for it to be coupled with the HAEM part of the hemoglobin. Normal adult blood. Remember we just said now that we have 3 types of hemoglobin. That normal hemoglobin that an individual has, and it's this type of these different types of hemoglobin function in individual at different stages of individual life. So we have the embryonic hemoglobin that is actually predominant at an embryonic stage, while we have fetal hemoglobin that is predominant at fetal stage, and we have the adult hemoglobin that is predominant at adult stage of an individual. And we said there is a switch as the embryo is mature, is developing, as that is a switch from embryonic hemoglobin being the predominant form of hemoglobin to fetal hemoglobin being the adult, fetal hemoglobin being the predominant form of hemoglobin, and then the adult hemoglobin taking over at the later stage. But one thing you should know that it's not as if there is a switch somewhere, but as the embryo is developing, the rate at which the embryonic hemoglobin is being synthesized and produced will reduce, while the rate at which the fetal hemoglobin is produced is increasing. So at a stage, the fetal hemoglobin will just be the predominant hemoglobin being engaged in carrying oxygen from the lungs to the tissue. At the same time, remember in red cell production, we have these 3 different stages. If you remember, we have the mesoblastic stage, we have the hepatic stage, and we have the myeloid stage. If you remember your lecture on erythropoiesis very well, and hematopoiesis very well, you remember that hemoglobin, red blood cells and other blood cells are produced, based on the site of production. You said that 3 stages, and this is also dependent on the age of individual. At mesoblastic stage, it is at the yolk sac predominantly responsible for production of these red cells. While hepatic stage, you have the liver and the spleen, and then the adult stage, the myeloid stage, you have the bone marrow being responsible. So what am I saying here? What I'm saying in essence is that normal adult blood at adult stage, normal adult blood contains small quantities of 2 other hemoglobin. So apart from the hemoglobin A, which is adult hemoglobin that we're aware of, in normal adults, we have other 2 forms of hemoglobin present in small quantity, fetal hemoglobin and hemoglobin A2. Now, I want to mention this thing here. When a child is born with predominantly fetal hemoglobin. As from 5-7 months in the womb, fetal hemoglobin is actually in control of doing the function of hemoglobin. But adult hemoglobin is also being produced gradually. So when a child is born with predominantly fetal hemoglobin with little adult hemoglobin being present. And as the child is growing from 1 month, 2 months, 3 months, there's a gradual decline in number or quantity of fetal hemoglobin being produced and an increase in adult hemoglobin. And that is why when you are doing hemoglobin electrophoresis using cellulose acetate and these other methods that don't actually quantify, it's not always good to do hemoglobin electrophoresis using cellulose acetate method for a child that is below 6 months, because when you do that, most times what you are actually isolating at that point is fetal hemoglobin. So as the child is growing, the rate at which the fetal hemoglobin is synthesized is being reduced, while the rate at which the adult hemoglobin is synthesized is also increasing until the adult hemoglobin completely takes over the function of carrying oxygen from the lungs to the tissue. That is the hemoglobin function. So in adult stage, you still have this little amount of fetal hemoglobin still being persistent in adult stage. In people who are sicklers, if it persists, somehow it is of benefit for them. Call it persistent fetal hemoglobin. In some hemoglobinopathies, you see fetal hemoglobin persisting. And there's also another form of hemoglobin that you can also find in adults, which is hemoglobin A2. In some hemoglobinopathies, you also see this hemoglobin A2 being increasing, like in thalassemia syndrome. All these things, I'm just mentioning them in passing here. They are abnormal cases, which we are going to discuss in later lecture. But for today, a normal hemoglobin, in a normal individual, you can have small amount of fetal hemoglobin being present, in addition to small amount of hemoglobin A2, and in addition to predominantly adult hemoglobin. And this fetal hemoglobin, like I said, fetal hemoglobin and hemoglobin A2, the difference is actually in the globin chain, containing either alpha or delta chain, while the normal adult hemoglobin, in addition to alpha chain, they contain beta globin chain. The major switch from fetal to adult hemoglobin occurs from 3 to 6 months after birth. The major switch from fetal to adult hemoglobin occurs from 3 to 6 months after birth. So what I just said earlier, and I said normal hemoglobin in adult blood. The normal hemoglobin you can find in adult blood are hemoglobin A, hemoglobin A2, and hemoglobin F. And I said here that each molecule of normal hemoglobin consists of 4 polypeptide chains, each with its own heme group. I said that heme group is actually constant in this hemoglobin. The difference is just in the structure of their globin chain, with hemoglobin A containing 2 alpha globin chains and 2 beta globin chains, while hemoglobin A2 consisting 2 alpha globin chains and 2 delta globin chains. Fetal hemoglobin contains 2 alpha globin chains and 2 gamma globin chains. And then the percentage, the normal percentage that you can see in normal adults of this hemoglobin is hemoglobin A. You have 96 to 98% being present in normal adults. So that is why it is predominant adult hemoglobin you find in adults. Now you have hemoglobin A2 being occurring at about 1.5 to 3.2%. Hemoglobin A2 in adults, you have it around 1.5 to 3.5%, while hemoglobin F, you have quantity being present in normal adult at 0.5 to 0.8% of the total hemoglobin. So you have hemoglobin A taking over about 96 to 98% of total hemoglobin of an adult individual. Hemoglobin A2 having 1.5 to 3.2% of total hemoglobin content of an individual, while fetal hemoglobin having 0.5 to 0.8% of total hemoglobin content of an individual. In a situation where you have hemoglobin A2 decreasing and hemoglobin A, a situation where you have adult hemoglobin decreasing and then hemoglobin A2, the quantity increasing more than this, or fetal hemoglobin increasing more than this, that means there are underlying issues. And this can be used in diagnosing some hematological ailments like thalassemia and also sickle cell anemia, those that have persistent fetal hemoglobin. Heme synthesis. The normal hemoglobin synthesis requires an adequate supply of iron and normal production of both protoporphyrin and globin. Normal hemoglobin synthesis requires an adequate supply of iron and normal production of protoporphyrin and globin. Now, we said that hemoglobin is made up of 2 basic parts, the heme part and the globin part. We've looked closely at the globin part. Now, we are going to be discussing the heme part. The heme aspect of hemoglobin is made up of protoporphyrin ring and iron. Protoporphyrin synthesis is initiated in the mitochondria with the formation of delta amino levulinic acid from glycine and succinyl coenzyme A. Protoporphyrin synthesis is initiated. Remember I said that heme is made up of iron and protoporphyrin ring. Now, the protoporphyrin synthesis is initiated in the mitochondria with the formation of delta amino ribulonic acid from glycine and succinyl coenzyme A. Now, synthesis then moves to the cell cytoplasm for the formation of porphoblinogen, uroporphyrin, and coproporphyrin. We are going to discuss these things in detail as we proceed. The assembly of protoporphyrin ring is carried out by the mitochondria, after which iron is incorporated under the control of the cytoplasmic enzymes, ferrochelatase, to form HAEM. Now, what we are trying to say here is for HAEM to be produced, protoporphyrin is first of all produced, and then iron is incorporated into it. Production of protoporphyrin starts in the mitochondrial part of the cell, extends to the cytoplasmic part of the cell, and then back to the mitochondrial part of the cell, and then back to cytoplasmic part of the cell. That's why we said that HAEM synthesis is largely done in the mitochondria, while globin synthesis is in the polyribosomes found in the cytoplasm. The final assembly of hemoglobin molecule occurs in the cell cytoplasm. Small amount of iron, protoporphyrin, and free globin chains remain after hemoglobin synthesis is complete. The iron is stored as ferritin, whereas the excess porphyrin is complex to zinc and is removed out of the system. HAEM synthesis. Let us now take a detailed look at how protoporphyrin is produced. HAEM synthesis starts with the condensation of glycine and succinyl coenzyme A under the action of a rate-limiting enzyme called delta amino ribulonic acid synthase. Delta amino ribulonic acid synthase. This reaction gives rise to delta amino ribulonic acid. So condensation, the first step in protoporphyrin ring synthesis is the condensation of succinyl CoA and glycine in the presence of an enzyme called delta amino ribulonic acid synthase to form delta amino ribulonic acid. In this particular reaction, vitamin B6, also known as pyridoxal phosphate, is used as a coenzyme. Vitamin B6 participated in this particular reaction as a coenzyme in the conversion of succinyl CoA and glycine to delta amino ribulonic acid in the presence of delta amino ribulonic synthase and pyridoxal phosphate vitamin B6 as coenzyme. A series of biochemical reactions after the formation of delta amino ribulonic acid, a series of biochemical reactions will follow. And this series of biochemical reactions involve two molecules of delta amino ribulonic acid will condense to form a pyrrole called porphoblinogen. Porphoblinogen, that is PBG for short. 4 molecules of porphoblinogen will condense under the influence of porphoblinogen deaminase and uroporphoblinogen co-synthase to form the tetrapyrrole ring compound called uroporphyrogen 3. The UPG3 is then converted to coproporphyrogen, CPG. This CPG then changes to protoporphyrogen and then protoporphyrin. which ultimately combines with iron in the ferrous state under the influence of an enzyme, ferrochelate, to form HAEM. Iron is brought to the developing red cell by a carrier protein that is transferrin. If you remember during our iron metabolism, we said transferrin is the protein that is responsible for transporting iron in the blood, carrying iron to various tissues where it is needed. Transferrin, which attaches to special binding site, that is transferrin receptor, on the surface of the cell, transferred iron into the cell through the process of endocytosis, and iron is incorporated into the protoporphyrin that is already being formed. Transferrin releases iron and returns back to the circulation. Each molecule of HAEM combines with globin chain. Globin chain is being formed simultaneously as the protoporphyrin ring is being formed, and iron is being brought in to be incorporated into the protoporphyrin ring to form HAEM, which then combines with globin already formed in the cytoplasm to form what? Hemoglobin. A tetramer of 4 globin chains, each with its own HAEM group, in a pocket is formed to make up a hemoglobin molecule. Let us take a closer look, diagrammatically look at what we have been explaining so far. The formation of protoporphyrin ring starts with condensation of succinyl-CoA, that is succinyl coenzyme A, adding with glycine to form delta amino levulinic acid. This delta amino levulinic acid is formed as a result of this enzyme being part of this reaction. This enzyme is a rate-limiting enzyme in this particular reaction. Delta amino levulinic acid synthase. And we also say that in this particular reaction, pyridoxal phosphate, which is vitamin B6, also is utilized here as a coenzyme. So synthesis of HAEM starts with what? Formation of protoporphyrin. And this formation of protoporphyrin starts in the mitochondrial part of the cell. And the first reaction that initiates the formation of protoporphyrin is condensation of succinyl plus glycine to form what? Delta amino levulinic acid. Now, when the delta amino levulinic acid is formed, this now moves to the cytoplasm to form profibrinogen. The profibrinogen now condensed and then acted upon by profibrinogen deaminase and uroprofibrinogen co-synthase to form uroprofibrinogen. This now gives rise to another compound, coprofibrinogen, which is formed in the cytoplasm. That is the CPG. The CPG formed in the cytoplasm moves back into the mitochondria, and protoporphyrin is formed. Protoporphyrin gives rise to protoporphyrin, and protoporphyrin, in the presence of an enzyme, ferrochelate, and iron that is already being, remember that iron is either gotten from stored iron that is stored as ferritin, or the iron we eat through our diet, or recycled iron from old red blood cell that has been destroyed. The iron is incorporated into transferrin and carried to the surface of the red cells, where there is transferrin receptor that collects this iron from transferrin and internalizes it into the red blood cell. And in form of ferrous iron, it is incorporated into protoporphyrin ring that has already been formed. The protoporphyrin ring that's already been formed, when iron is incorporated into it, in the presence of ferrochelate, give rise to HAEM. The HAEM now combines with globin chain that's already been formed from different alpha globin and beta globin chain in the case of hemoglobin A to form alpha beta globin chain. And this combines with the heme that's already been formed to form hemoglobin. So hemoglobin is made up of heme and globin, and heme in itself is the production synthesis of heme is done largely in the mitochondria, while part of it is done in the cytoplasm and ends up back in the mitochondria. And it starts with the production of protoporphyrin, which we have already discussed, stated. This is also the diagrammatic explanation of what I have just said, where you have the heme, the ion, where you have the ion, you have the ion being brought into the cell and being incorporated into the protoporphyrin has already been formed. And you have inside the mitochondria, this is the mitochondria, you have glycine and succinic coenzyme A coming together in the presence of delta aminolevulinic acid synthase to form beta aminolevulinic acid, which leaves the mitochondria into the cytoplasm to form porphobrinogen and then uroporphyrin, CPG, and then back into the mitochondria to form protoporphyrin and then protoporphyrin, and then ion being incorporated into protoporphyrin ring to form heme, and then heme coming back to combine with globin that has been formed from the polyribosomes, that is from amino acids in the polyribosomes, either as alpha chain, globin chain, or beta globin chain, or delta globin chain, or gamma globin chain, depending on the hemoglobin that is being formed. And this joins with the heme group to form what? Hemoglobin. This is the structure of hemoglobin with the protoporphyrin ring, and you have ion at the center, and then globin chain. All this is just the protoporphyrin ring. You have the ion at the center, and then you have the globin being attached to it. And this is what? Hemoglobin molecule. And then heme consists of protoporphyrin ring with an ion atom at its center. The protoporphyrin ring consists of 4 pyro groups which are united by methane bridges. That is it. The hemoglobin containing protoporphyrin ring, ion, and globin chain. The protoporphyrin ring and ion makes up the heme aspect of the hemoglobin. Control of heme synthesis. Synthesis of heme and globin is coordinated so that there is no significant excess of either. There is no significant excess of either. The entry of ion into the cell and its incorporation into heme are regulated so that the normal cell obtains sufficient ion for its needs and not more. So the synthesis of heme and globin is so organized, so well coordinated that there is no significant excess of either. So whenever you see protoporphyrin ring be accumulated in the system, it is only in pathological state. That means something is wrong somewhere, like that denies ion being brought in. So maybe in cases of iron deficiency, protoporphyrin ring is being formed, but no ion to be incorporated. So you have protoporphyrin ring accumulating in the system. That means it's in a pathological situation. Or you have the cases where you have sideroblastic anemia, where something is happening to the enzymes in the system, or in porphyrias, where something is happening, either inhibiting the rate-limiting enzyme, which is delta aminolevulinic acid, or vitamin B6 is not available to be used as a coenzyme, or in lead poisoning. In all these situations, is a pathological case where you have an interference in the well-organized system that brings ion, protoporphyrin, and globin together to form hemoglobin. The entry of ion into the cell and its incorporation into heme are regulated so that the normal cell obtains sufficient ion for its needs and not more. You know, when we did iron regulation, we talked about this, that the system is so coordinated that the iron is so regulated that the exact amount of iron is taken to where it's needed at the right time. So the incorporation of iron into protoporphyrin ring is done so that you don't have excess iron being around inside the mitochondria. Remember that free iron is toxic to the system. The individual components of hemoglobin synthetic pathway, like iron, free protoporphyrin, heme, monomeric globin chain, they are all extremely toxic to the cell. Therefore, the system is done in a way, the regulation is done in a way that none of these things is in excess for it to accumulate inside the cell to cause a harm to the cell. In a situation where you see iron accumulating in the cell, like in sideroblastic anemia, there is a underlying pathology. You have free protoporphyrin ring accumulating in the system. That is in lead poisoning and porphyrias and things like that. There is underlying pathology, which we will treat in subsequent lectures. The synthesis of globin must be very accurately matched with the synthesis of HAEM. The synthesis of globin must be well accurately matched with the synthesis of HAEM. Delta aminolevulinic acid synthase is the rate-limiting enzyme in the erythroid HAEM biosynthetic pathway. That particular enzyme is the rate-limiting enzyme in this particular pathway. Its activity reaches its peak at the polychromatic normoblast and then diminishes so that no activity is present in the mature cell. Interleukin-3, erythropoietin act as inducers for transcription of messenger RNA for delta aminorevulinic acid synthase and profibrinogen deaminase. Continued translation of globin chains from messenger RNA only occurs in the presence of adequate HAEM. When you don't have enough HAEM that can combine with globin, then the messenger RNA is deactivated. Reduced level of HAEM rapidly triggers the formation of the HAEM-regulated inhibitor, a kinase that interacts with the translation initiation factor and prevents translation of alpha and beta globin messenger RNA. The synthesis of HAEM itself is also regulated at many points and is particularly sensitive to the level of available iron. That's why when there's deficiency of iron in the system, you have ineffective erythropoiesis. The synthesis of HAEM itself is also regulated at many points and is particularly sensitive to the level of available iron. Through a well-characterized pathway involving iron regulatory protein, like we've discussed when we are discussing iron regulation, binding to iron-responsive elements in the messenger RNA transcript of ferritin, transferrin receptor, and delta aminorevulinic acid synthase, the pathway that regulates iron metabolism within the cell, the iron regulatory protein 1 and 2, and iron responsive element pathway. The level of intracellular iron thus controls the translation of RNAs involved in iron storage, ion transport, and HAEM synthesis. Remember, like I said before, iron is very important in erythropoiesis, in the formation of HAEM. When it is not available, HAEM cannot be produced. Protoporphyrin ring can be produced, but it accumulates. And that is why in iron deficiency, one of the tests that we do to answer for iron deficiency is to estimate protoporphyrin ring in the system. Estimation of protoporphyrin ring will help us to know, because when we do and we have accumulation of protoporphyrin ring, it then means that the body is deficient of iron. So estimation of protoporphyrin ring is one of the tests that we do to diagnose iron deficiency anemia. Red cell destruction. Let us now discuss hemoglobin catabolism. Hemoglobin catabolism. Red cell destruction usually occurs after a mean life of 120 days. The cells are removed extravascularly by macrophages of the reticuloendothelial system, especially in the bone marrow, but also in the spleen and the liver. Red cell metabolism gradually deteriorates as enzymes are degraded and not replaced until the cells become non-viable. But the exact reason why the cells die is still not very clear. Breakdown of red cell liberates iron. For this iron is recycled and reused again. It liberates protoporphyrin ring, which is broken down to bilirubin. It also replaces globin, which are converted to amino acid. So the breakdown of red blood cells liberates iron, protoporphyrin ring, and globin. After phagocytosis by macrophages, HAEM from senescent red cells is broken down by HAEM oxygenase to release the iron. As ferrous iron, it can then either enter ferritin when iron is released as part of hemoglobin catabolism. When iron is released, this iron is recycled. It can either enter back into the pool. Remember, transferrin is what carries iron in the blood. So it can be carried by transferrin and be transported back to ferritin and restored. Or it can be taken straight back by transferrin back to bone marrow and be reused to produce more red blood cells. The bilirubin, that is when protoporphyrin is broken down to bilirubin. And the small gas, carbon monoxide, is liberated as part of breakdown of protoporphyrin ring. The bilirubin circulates to the liver, where it conjugates with gluconides, which are excreted into the gut via bile and converted to either stachybilinogen or urobilinogen. The stachybilinogen is excreted in feces as stachybilin, while urobilinogen is excreted in urine as urobilin. Then a small fraction of protoporphyrin is converted to carbon monoxide and excreted via the lungs. Globin chain are broken down to amino acid, which are reutilized for general protein synthesis in the body. The globin chain is broken down to amino acid, which are reutilized for general protein synthesis in the body. Let us look at diagrammatic explanation of what we have just said so far. This is hemoglobin catabolism. Hemoglobin, during the destruction of hemoglobin is first of all broken down to HAEM and globin. HAEM and globin. Now, the HAEM part of hemoglobin is broken down to iron and protoporphyrin. The iron is incorporated into transferrin and then taken back to bone marrow, where it is utilized to produce more red blood cells. While protoporphyrin is broken down, so a small amount of carbon monoxide is released as part of breakdown of protoporphyrin and is expired in air. Now, the another one is bilirubin is released as a part of breakdown of protoporphyrin. And this bilirubin is conjugated in the liver to form by gluconolic acid to form bilirubin gluconides. And this conjugated bilirubin can now be either converted to urobilinogen taken out in urine or stachybilinogen taken out in feces. And this is why whenever there is increased destruction of red blood cells in the system, whenever there is any disease that causes increased destruction of red blood cells in the system, you see somebody, the individual, having what we call jaundice. Jaundice that we see is just due to excess destruction of red blood cells, because as part of destruction of red blood cells, bilirubin is produced. Normally, the liver will keep conjugating this bilirubin so that it can be removed through urine or through the feces as either sacchobilin or urobilin. But if the rate of destruction of this red cell is so high, then the liver cannot be able to conjugate it. So you see this bilirubin, which is yellow in color, being deposited in the tissues of the body. So you now see an individual having yellowness of the eyes, the palm, and then you look at some parts of the skin being yellow. You have the globin part of the hemoglobin is that now broken down to amino acid, which is now reutilized to synthesize the proteins that we have in a system. So that is, in a nutshell, the catabolism of hemoglobin, breaking down to these two basic parts, heme and globin, and the heme being broken down to iron and protoporphyrin ring, and then the iron part of it is reused, either stored in ferritin, is incorporated into transferrin, which is the transport ion, either stored as ferritin or reused in the bone marrow to produce more red cells. The protoporphyrin ring is broken down to bilirubin and then down to conjugated in the liver to give rise to urobilinogen and sacchobilinogen, which is excreted out in urine as urobilin or feces as sacchobilin. A small amount of carbon monoxide is also produced as a result of protoporphyrin ring degradation. And then globin is broken down to various amino acids, and then it is utilized to produce other proteins in the system. Thank you very much. For further reading, you can use any of these resources. You can use any of these resources. Thank you very much. I know that you have learned one or two things from this lecture. I have also enjoyed it. Please don't hesitate to send in your questions. Don't hesitate to send in your questions. We will be glad to answer them. Remember, you can drop your questions on the comment section of this lecture, or you can send it in through our email, scholarlywoman20@gmail.com. For further readings, you can use any of these textbooks, and also keep a date with us for the next lecture. Thank you very much. Thank you, and thank you.